How do muscles contract? Muscles are composed of bundles of muscle fibers, which are single large cells formed by the fusion of many individual cells during development. The cytoplasm of these muscle cells consists of myofibrils. These myofibrils are cylindrical bundles of two types of filaments: thick filaments of myosin, which look like this right here, and thin filaments of actin, which look like this. Each of the myofibrils are organized into long chains called sarcomeres, which give the muscle the striated appearance, which is long lines that look like this that are visible on the muscle. The sarcomeres have a fairly complex structure with distinct regions. Each end of the sarcomere is defined by the Z-line, which is shown here and here. Within each sarcomere are dark bands alternated with the light bands. The dark bands are called A bands, and light bands are called I bands. The I bands only contain actin filaments, which are these right here, and the A bands contain myosin filaments, which are shown here in pink. The middle region of the sarcomere is called the H zone, and it only contains myosin filaments. The H zone is this area right here. Inside all muscle fibers are motor neurons. These motor neurons are the starting point of muscle contraction. All motor neurons have similar structures. The cell body contains the nucleus and maintains the function of the neuron. Cell body is shown right here. The dendrites are part of the neuron that take in the information from the brain and pass it into the cell body. So the information comes into these ends of the cell body right here, which are called dendrites. After the information is passed into the cell body, it continues to move through into the axon. From here, it travels through the axon until it reaches the motor end plate. So the information comes into the cell body and through into the axon, where it comes down here into the motor end plate. And the motor end plate is usually attached to the muscle, which is shown right here. The information that is passed from the brain to the motor neuron is called an action potential. Before the muscle is able to contract, the nerve signal must stimulate the muscle. It does this in the process of synaptic transmission. Once the action potential reaches the motor end plate, calcium ions are released into the motor end plate. So the action potential travels into the motor end plate through here, and because of this, calcium ions are released into the other areas of the motor end plate here. The calcium sends a signal to the synaptic vesicles that are located in the motor end plate. These right here are the synaptic vesicles. The signal tells the vesicles to bind with the membrane and release the neurotransmitter acetylcholine into the synaptic cleft. So these vesicles move down into this area and they bind with the membrane, and once this happens, the neurotransmitter acetylcholine is released into the synaptic cleft, which is this area between the motor end plate and the muscle. The acetylcholine diffuses across the synaptic cleft to find the specific receptor site on the postsynaptic cell. So the acetylcholine is binding to the postsynaptic cell in these areas right here, which is their specific receptor site. The acetylcholine opens the receptor site, which allows sodium ions to enter the muscle, creating another action potential, which helps to cause the muscle contract. Cholinesterase is an enzyme that immediately starts breaking down the acetylcholine when it is released into the synaptic cleft to prevent too much of a chemical reaction. Once the action potential travels into the new muscle cell from the synaptic transmission, we wonder how it gets to all the other muscles throughout the cell quickly. But we know this happens through a series of pathways called T-tubules. The T-tubules are located in muscle fibers and can pass action potentials quickly to all myofibrils in a muscle fiber. The T-tubules look like this. They're vertical tubes located in the muscle fiber. The action potential is transmitted from the sarcolemma, which is the cell membrane, shown here and here, to the sarcoplasmic reticulum, which then releases calcium ions into the sarcoplasm. The sarcoplasmic reticulum are these lines here that are horizontally in the cell fiber. Muscle contraction can be compared to using oars on a rowboat moving you across a lake. To move across the lake, the oar must be placed into the water and pulled backwards. At the end of the stroke, the oars are lifted out of the water, pushed forward, and then placed back into the water for the next stroke. The movements of the oars propel the boat across the water. The movements of a muscle contraction are very similar to these movements of the oar, and it is called the sliding filament theory. Another thing that the sliding filament theory can be compared to is a Chinese finger trap. As the fingers of the person are pulled back, the trap contracts and basically traps the fingers inside. But once the person moves their fingers forward, it looses and the person can pull their fingers free. The calcium ions released from the action potential travel to the myofilaments actin and myosin. The attraction between the actin and myosin is so strong that it needs to be regulated to prevent unwanted muscle contraction. The two proteins, troponin and tropomyosin, are used to regulate this. The calcium ions bind to the troponin, which cause it to move and uncover the myosin binding site, which is shown here. Once these sites are exposed, the myosin heads bind to the actin, forming a cross bridge. The ATP energy stored in the myosin head causes it to cock back using the power stroke. So myosin normally looks like this, but once the ATP energy is used and the power stroke is used, the myosin head cocks back and is in this position. This movement can be compared back to a rower with an oar. This shortens the sarcomere as the actin and myosin move in opposite directions. This process is occurring in every sarcomere along the length of the muscle, which causes it to contract. Another ATP then stimulates the myosin head to release the actin and move back to its original position, which is relaxed. This process repeats itself until the calcium ions are removed and the tropomyosin-troponin complex covers the myosin binding sites again.